Hello guys, now in this video let's discuss about renal physiology. Initially I will be mainly concentrating on basics and wherever possible let's try to integrate with patho and pharma. Now let's start with the basics. So what are the functions of the kidneys? Guys, you all know that kidneys are the ones which are mainly involved in the process of excretion. Whatever the nitrogenous waste products which are getting produced in the body, that is the blood urea nitrogen and the creatinine. So these waste products need to be eliminated out of the body. And who is going to take care about this function? It's the kidneys. So nitrogenous waste product excretion. So that's the major function of the kidneys. So what are the other functions? EPO secretion. What exactly is EPO? Erythropoietin. So erythropoietin production and secretion is also going to be taken care by the kidneys. Now in the kidneys, which cells are going to produce this erythropoietin? We all know erythropoietin is the one which is helpful in the production of more and more RBCs. So erythropoietin helps in the production of RBCs. Erythropoietin is getting produced from the kidneys. In the kidneys where? Which cells? So please see guys, peritubular capillaries. So peritubular capillaries are the ones in the kidneys which are helping in the production of erythropoietin. Okay. Apart from that, renin. Renin secretion is also going to be taken care by the kidneys. Now we will discuss in detail about what renin is going to do. That is the renin angiotensin aldosterone system. But for now, just remember renin is also going to be synthesized and produced from the kidneys. Now in the kidneys, which cells? It's the JG cells. Okay, JG cells means juxtaglomerular cells in the kidneys, or I should say juxtaglomerular cells which are present in the nephrons are the ones which are going to produce the renin, and that renin will do a cascade of reactions which will help in increasing the BP. That we will discuss later. Now, so we have discussed three functions: nitrogenous waste product excretion, erythropoietin production and renin production. Apart from that, activation of vitamin D. Guys, vitamin D is something which is actually produced in at the level of skin itself. Okay, so with the help of the sunlight, the cholesterol whatever is present in the underneath the skin is going to be converted into the vitamin D, but that vitamin D is highly inactive in form. That inactive vitamin D need to be activated. So vitamin D activation, where it's going to take place? Vitamin D activation is going to take place in the kidney. Again, where? In the PCT of the nephrons. PCT means proximal convoluted tubules. In the proximal convoluted tubules of the nephrons, the inactivated vitamin D will be activated. So activated vitamin D helps in the absorption of calcium and phosphorus. We will discuss in detail about how the vitamin D synthesis will happen later on. Now, the last function of the kidneys is acid-base balance. Okay, all the time the acids and bases in the body needs to be balanced. Otherwise, the person will suffer with alkalosis or acidosis. So acid-base balance, for this regulation of acid-base balance, there are many levels of systems which are present in the body. Like lungs can be having a role in regulating the acid-base balance. Kidneys are having a role in maintaining the acid-base balance. Buffer system is there. There are many levels of regulation of the acid-base balance, but one of the important, not major, but one of the important level at maintaining the acid-base balance is done by kidneys also. Okay. Now let's see in detail about how vitamin D is activated. We have seen vitamin D activation is going to happen in the PCT of the kidneys. Now let's see how actually it happens. Guys, let's start with the sun. Okay. Now in the sun rays, we are particularly interested with the UVB rays. Okay, the ultraviolet B rays that are going to fall onto the skin. Now then what happens? When this ultraviolet B rays, when they fall onto the subcutaneous fat, there the cholesterols, which cholesterol? Cholesterol is going to be converted into 7-dehydroxycholesterol. Now this 7-dehydroxycholesterol, whatever is there under your skin, with the help of UVB light, it will be converted into cholecalciferol. This is the first step. Where first step is happening? At the level of skin. So cholecalciferol is there. Now this cholecalciferol is going to come to the liver. Now in liver what happens? Now for the first time this cholecalciferol is going to be hydroxylated on 25th carbon position. So now this cholecalciferol is going to be converted into 25 hydroxy cholecalciferol. Okay, so on 25th carbon position hydroxylation is going to be happening. So that's why we kept hydroxy group. So 25 hydroxy cholecalciferol is now there in the liver. Now what happened to this 25 hydroxy cholecalciferol? Now this one is going to come to the kidneys, Then what happens? Now in the kidneys, in the PCT of the nephrons, this 25 hydroxy cholecalciferol is getting one more hydroxylation on carbon number 1. So now it's becoming 1,25 dihydroxy. Okay, there are two hydroxylation. One hydroxylation is there on the 25th carbon and the second hydroxylation or the final hydroxylation is present on the first carbon. So 1,25 dihydroxy cholecalciferol is there. Now this 1,25 dihydroxy cholecalciferol is the real vitamin D. So it's the active form of vitamin D3. Okay, active vitamin D3. Okay, so this is what's happening in the PCT of the nephron. But the important MCQ is that, see, this step is regulated by, now this very important step in the PCT, it's under the control of a enzyme known as alpha-1 hydroxylase. See, this alpha-1 hydroxylase, it is present in the PCT of the nephrons. Now this alpha-1 hydroxylase is the real one who is going to convert 25 hydroxy cholecalciferol into 1,25 dihydroxy cholecalciferol. Also, if you can remember, please keep this point in mind. See, this alpha-1 hydroxylase, okay, alpha-1 hydroxylase, this enzyme is under the control of which hormone? There is one more hormone which controls this step. What is that? Parathyroid hormone. So parathyroid hormone will control alpha-1 hydroxylase activity. Now this alpha-1 hydroxylase, what it is doing? It's going to convert the inactive vitamin D into active form of vitamin D. Now what is this active vitamin D going to do? This active vitamin D is going to rush to the GIT mucosa. Now in the GIT mucosa, it's going to help in the absorption of, it helps this vitamin D, it's going to help in the absorption of both calcium and phosphorus. This is a very important MCQ. Both the calcium absorption as well as phosphorus absorption. Okay, both calcium and phosphates, both are going to be absorbed in the GIT with the help of vitamin D. Okay, if you know this much, this is more than enough for your exams. So this is how vitamin D activation takes place. Now after this, let's talk a few important points about the RAAS pathway, which is also known as the renin angiotensin aldosterone system or renin angiotensin aldosterone axis. So what actually happens in this RAAS? Guys, at the end of the day, one important point you need to keep in mind. What is the function of RAAS? The one important function of RAAS is that all the time it wants to increase the BP. The RAAS function is that it increases the BP in the person. Once RAAS pathway is activated, the BP of the person will be increased. Now you should tell me when the RAAS pathway will be activated. So whenever the person is going into hypotension, whenever the BP of the person is getting decreased, in that conditions, RAAS pathway will be activated. Now activation of RAAS pathway will increase the BP. How, sir? That we will discuss now. See, this is the starting point. So we are going to start with a condition where the BP of the person is getting decreased. Okay, the BP of the person, our patient's BP is getting decreased. This is the first stimulant. And also remember, RAAS pathway will be activated in those persons whose sodium levels will also be decreased. So hyponatremia or hypotension, they are the stimulants for the activation of RAAS. There is also one more stimulant which we will discuss in the next slide. But for now, see, BP decreased, hypotension, and decreased sodium, that is hyponatremia. And when the sympathetic system in the person is also activated, that also will be a trigger for the activation of RAAS. So there are three stimulants, I should say there are three triggers for the activation of RAAS. What are the three triggers? Hypotension, hyponatremia, sympathetic activation in the body. All of them will activate the RAAS pathway. So how they are going to activate the RAAS pathway, we will discuss. Whenever the BP of the person is going down, or whenever there is the hyponatremia, the JG cells in the nephrons, there are certain cells which are known as the JG cells. I will show you where exactly those JG cells are, but for now, just keep this point in mind. Now the JG cells, they can sense the hyponatremia and the hypotension. Now what they are going to do? Now they are going to release a hormone called as renin. Okay, so JG cells are going to produce renin. Now what this renin is going to do, sir? Now this renin is present in the blood. Now JG cells are going to release the renin. Now this renin is coming into the circulation. Okay, now this renin is present in the blood. Now what happens, sir? What is the function of this renin? This renin is going to convert this angiotensinogen. This angiotensinogen is already there in the blood. This angiotensinogen is produced by the liver. That's a total different thing. Angiotensinogen is something which is already present in the blood. Now on this angiotensinogen, renin is going to work. Now what happens when renin acts on angiotensinogen? It will be converted into angiotensin 1. Later what happens, sir? This angiotensin 1, it goes to the lungs. Okay, now it's coming to the lungs. In the lungs what happens? With the help of an enzyme called as ACE, angiotensin converting enzyme. This angiotensin converting enzyme is present in the pulmonary capillaries. Now this angiotensin converting enzyme which is present in the pulmonary capillaries, there what it's going to do, it's going to convert angiotensin 1 into angiotensin 2. Now the question is, conversion of angiotensin 1 into angiotensin 2 happens in lungs with the help of enzyme, angiotensin converting enzyme. Okay. Now what this angiotensin 2 is going to do, sir? See, this angiotensin 2 directly, it's a potent vasoconstrictor. Okay, this angiotensin 2, it can act on the blood vessels and it will cause vasoconstriction. So when vessels are getting constricted, what happens? BP automatically increases. BP will definitely increase. So this vasoconstriction will cause increase in the blood pressure. So our target is getting achieved or not? Yes. The purpose of RAAS pathway is to increase the BP. How it's getting increased? In the lungs, angiotensin 1 is going to convert into angiotensin 2. This angiotensin 2 is a potent vasoconstrictor. So because of the vasoconstriction, when vessels are getting constricted, BP will be increased. This is one way. And this angiotensin 2, there is also one more way. By which it can increase the BP. So what is that, sir? Please look at here. This angiotensin 2, it's going to act on the adrenal gland which is present over the kidneys. Now this angiotensin 2, it will act on the adrenal gland. We all know that in the adrenal gland there are regions like cortex and medulla. Adrenal cortex is further divided into, we have already discussed these things in the endocrinology. Cortex was further divided into zona glomerulosa, fasciculata, reticularis. All these are the parts of adrenal gland. Okay, even if you don't know, don't worry. I am simply saying you that this angiotensin 2, it's going to act on the adrenal gland. Which part of the adrenal gland? Zona glomerulosa. Okay, on the zona glomerulosa, it's getting acted. Now what happens? Now aldosterone is going to be produced. Okay, aldosterone is going to be produced and released out of the adrenal gland. Now this aldosterone, which is a steroid, it's a mineralocorticoid. Okay, aldosterone is a mineralocorticoid. What does I mean by mineralocorticoid? See, it's a steroid hormone which is associated with mineral metabolism, I should say. So what is that mineral metabolism, sir? See, this aldosterone, whatever is coming out, it's going to reabsorb the sodium. The kidneys are going to reabsorb more and more sodium under the presence of aldosterone. So simple, whenever there is aldosterone, more sodium is going to be reabsorbed. When more sodium is coming into the body, what happens? Automatically BP increase. We all know, right? When if you take more salt, BP will increase. So aldosterone, it's going to come back to your blood because of more reabsorption in the kidney. From the urine, more and more sodium is getting reabsorbed. So BP will increase. So this is a very simplistic way of RAAS pathway. At the end of the day, RAAS pathway all the time wants to increase your BP. What are the stimulants? hyponatremia, hypotension and sympathetic activation. Okay, this is what you should know. Now, see, this is MCQ. What are the stimulus for the RAAS activation? I have already discussed hypotension, hyponatremia and sympathetic activation. Now, here important point you need to keep in mind is, see this beta 1. What is this beta 1, sir? You can ask me. Please look at here, guys. See the JG cells, these JG cells, where they are present? They are present at the level of nephrons. For example, right now, my sympathetic system is getting activated. Now, if sympathetic system is getting activated, it's going to produce the catecholamines, it's going to release norepinephrine and epinephrine. Now, that norepinephrine and epinephrine should have to come and act on the JG cells. So, for that, there should be some receptor which is present at the level of JG cells. So, what is that receptor, sir? Beta 1 adrenergic receptor. On the juxtaglomerular cells, on their surface, there is this one receptor called as the beta 1 receptor. Now, on the beta 1 receptors, these catecholamines will come and act here. Okay, epinephrine, norepinephrine, they will come and act here. So, that is the sympathetic activation. Now, JG cells will be activated and again they will produce the same pathway, renin, angiotensinogen to angiotensin 1, angiotensin 1 to angiotensin 2, angiotensin 2 will help in the production of aldosterone, the same pathway which we have already discussed. Okay. So, what is the sympathetic receptor which is present on the JG cells? Beta 1 adrenergic receptor. Okay. Now, here I just want to integrate with the pharma. For example, if you want to inhibit the RAAS pathway, for example, I am the patient who is having hypertension. Now, hypertension, how can you decrease the BP in this patient? There are many ways. One important way where we can correlate our RAAS pathway is that renin inhibitors, using renin inhibitors like aliskiren. Okay, aliskiren. What kind of drug is this aliskiren? Aliskiren will inhibit directly renin. So, whenever renin is inhibited, there is no way that angiotensinogen will be converted into angiotensin 1. If there is no angiotensin 1, there is no angiotensin 2. If there is no angiotensin 2, there is no aldosterone. No BP will be increased. Okay. So, renin inhibitor is aliskiren. Now, let's discuss a few more basic physiological points regarding the renal system. Renal circulation. The name itself is that how much blood is going, how much plasma is going, how much blood is getting filtered. All these points now we will see. Okay, these are direct single liner kind of questions. RBF. RBF means renal blood flow. How much blood is going to the kidneys per minute? For both the kidneys, not one kidney, both the kidneys. How much blood is going for both the kidneys per minute? It is 1.2 liters. Okay. So, what is this? 20%. 20% means out of 5 liters. See, what is the normal cardiac output? The normal cardiac output is 5 liters. Out of 5 liters, almost 1 liter, 1.2 liters is going to the kidneys, which means 20%, right? 20, 20, 20, 20, 20. All 20s, all 5 20s, 100. So, how much blood is going to kidneys? 1.2 liters. So, 1.2, or I should say simply take it 1 liter. 1 liter out of 5 liters is going to kidneys. That is 20%. 20% of the cardiac output is getting diverted to the kidneys for the process of filtration. Guys, please know this. Actually, this 1.2 liters is what? This is the blood. We all know that blood is nothing but a mixture of plasma and cells. Okay, the blood is nothing but a combination of plasma, that liquid component, and the packed cells. Now the cells includes what? Cells include RBC, WBC, platelets, all these things. Now let me ask you one thing. Are your RBC, WBC, platelets, are they going to be filtered? Actually no. So from the blood, if you take the cell component away, what we are left with? Only liquid component. So this liquid component is the one, right, which is having all the waste products. So only we have to take this liquid component into account. So how much plasma is going to the kidneys? Now we all know usually 60% will be plasma, 40% will be the cells volume. Usually I'm just saying you a broad value. So out of this 1.2 liters, 625 ml will be the liquid component. That is a plasma. The renal plasma flow is 625 ml. This is the plasma only liquid. We have taken out the cells. Now, what is the GFR? Glomerular filtration rate. Means per minute. How much amount of filtration is happening at the level of glomerulus or the nephrons? How much is getting filtered, sir? See, 625 ml of plasma is going to both the kidneys for the process of filtration. I am asking you, this entire 625 ml will be filtered? No, it's not going to happen. So how much is going to happen? Out of this 625 ml, only 125 ml will be filtered. Okay, so 625 ml is coming to the both the kidneys. But out of the 625 ml, only 120, the kidney is having a capacity to filter 125 ml. That's a glomerular filtration rate per minute. Okay. Now, let's discuss one more formula. What is this? Filtration fraction. See, fraction, how much is coming and how much is getting filtered? Now, see, the filtration fraction formula is GFR, okay, GFR by renal plasma flow. What is the normal GFR value? GFR value is 125 ml. So, this much is getting filtered. And what is the renal plasma flow? Renal plasma flow is 625 ml. So, 125 by 625 is somewhere around 20%. So, if someone asks you what is the normal filtration fraction, the normal filtration fraction is 20%. It's 20%. Okay. So, these are some important formulas which you need to keep in mind. Now, renal plasma flow is estimated by, guys, this is a single liner kind of question which previously asked. Just for now, remember, it is the para-aminohippuric acid. Para-aminohippuric acid is the one which we use to know what is the renal plasma flow. See, we know, theoretically we know, the renal plasma flow is 625 ml. There is no doubt. But in studies, if you want to do an experiment kind of thing, how you will do it? See, by injecting para-aminohippuric acid into the person, we will estimate the renal plasma flow. Okay. So, this is you need to keep in mind.